Scientists are growing tiny pieces of human brain-like tissue in the lab, and the story is more complicated than the headline. You may have seen them called mini-brains. The technical name is brain organoids or neural organoids. They start with stem cells, cells that can be guided back into a flexible state, then nudged toward becoming nerve cells and support cells. In the right 3D environment, those cells do something strange. They self-organize. They do not become a full brain. They do not have a body, blood vessels like ours, sensory organs, memories, or the architecture that makes a human mind. But they can resemble pieces of early brain development closely enough that scientists can watch human neural tissue grow in ways that would be impossible inside a living embryo. That is why organoids matter. A mouse brain is useful, but it is not a human brain. A flat dish of cells is useful, but it cannot capture the same 3D interactions. A brain organoid sits in the middle, imperfect, tiny, messy, but alive with human biology. The moment that made the public look twice came in 2019. Researchers at UC San Diego reported that cortical organoids could develop organized electrical activity. The patterns were not thoughts, they were not proof of awareness, but they were brainwave-like signals that, in some measurements, resembled activity seen in very early premature infants. That finding did two things at once. It showed how powerful the model had become, and it made the ethical questions harder to ignore. Fast forward to 2026. Northwestern University and collaborators reported a soft, three-dimensional electronic mesh that can wrap around a living neural organoid. Older devices were mostly flat, so they could only listen to a small part of a round, millimeter-scale tissue. The new shape-conforming framework can cover most of the organoid surface and place hundreds of tiny electrodes around it. That means researchers can map signals across nearly the whole network instead of sampling one small patch. In one version, the device covered about 91% of the organoid surface and used 240 addressable microelectrodes. It can record activity. It can also stimulate selected regions. In plain English, scientists are learning how to listen to these mini brain models and how to talk back. The medical promise is huge. Organoids can be made from a patient's own cells, which means they may capture parts of that person's genetics. Researchers can use them to study early brain development, rare genetic disorders, epilepsy-like activity, autism-related pathways, drug responses, infections, and neurodegenerative disease. In April 2026, Johns Hopkins researchers described brain-like organoids made from blood samples from people with Alzheimer's disease. Those models showed disease-related differences in proteins involved in communication, inflammation, and other pathways. A model like that does not replace a human brain, but it can give researchers a living test system that carries patient-specific biology. This is the optimistic version of the story. Fewer blind guesses, fewer animal experiments, better disease models, and maybe faster drug testing. If a lab can grow a tiny model of a patient's neural tissue, test several compounds, and see which pathway changes, that could eventually make parts of neuroscience more personal. But the hard question is where the line is. Current brain organoids are widely regarded as far too limited to be conscious. They usually model parts of brain tissue, not an integrated mind. Still, the field is moving toward more mature organoids, fused organoid systems called assembloids, vascularized models, transplantation experiments in animals, and interfaces with computers. Each step can be scientifically useful. Each step also raises new questions. Could a future organoid feel pain? What biological signs would tell us? Should there be limits on how long these tissues are grown, how complex they become, or whether they are connected to external devices? The NIH Brain Initiative has treated these as real neuroethics questions, not science fiction. The ISSCR stem cell guidelines also emphasize oversight, accurate public communication, and special attention when human neural models become more complex. So the honest answer is not that scientists have grown a conscious brain in a dish. They have not. The honest answer is more interesting. Researchers have built living human neural models that can develop circuits, fire coordinated signals, reveal disease biology, and now connect to electronics in ways that were not possible before. Brain organoids are not tiny people, but they are no longer just blobs of cells either. They are a new window into the human brain, and every improvement makes the science more powerful and the responsibility heavier. The breakthrough is not a mini-mind in a jar. The breakthrough is that we are finally getting a living, human-scale testbed for questions the real brain has kept hidden.